Namaste friends, welcome back to Biology Center for True Learning. In previous video, we discussed about the monohybrid cross. In this video, we will talk of dihybrid cross. Dihybrid cross. So it is also called inheritance of two genes. So this experiment specifically did by Mendel to know the influence of one pair of traits of characters on other pair of traits. That is the main intention. So let us see. So what is the dihybrid cross? Then what is the procedure involved in the dihybrid cross? Then what are the phenotype ratios and the genotype ratio received by the Mendel? And finally the conclusion given by the Mendel from his observations. I mean from this cross. So what is dihybrid cross? As the name indicates, the cross between two plants differing in two pair of contrasting, I mean opposite, contrasting traits. The meaning is very simple. So when the cross is made between two different plants differing in two pair of contrasting traits, this is called a dihybrid cross. In case of monohybrid cross, so Mendel only selected two plants differing in only one pair of contrasting traits, but in case of dihybrid cross, two pair of contrasting traits or two pair of opposing traits are selected for the experiment. Then, so what is the process, experimental process? So already I explained in the previous videos. So here I briefly explain them. The process involved in the cross or experiment. So it involves various steps. Step number one, selection of parents. Step number two, hybridization, that is cross pollination between parents. Step number three, selfing of hybrid. So whatever progenies we get in the F1 generation, so between them the self pollination is done, that is selfing of hybrids. So this is the simple process. So again Mendel used for the dihybrid cross also. So let us involve into the main aspect of the dihybrid cross. So it is better to go through the diagrammatic representation, then it is very easy. So before that I would like to tell you what are the characters. So what are the two characters selected by the Mendel, then what are the contrasting traits of each character. So first write down the characters selected by Mendel. The one is seed shape. The another one is seed color. Then here different traits are there. In case of seed shape, two traits. The one is round shaped seeds. The another one is wrinkle shaped seeds. The round shaped seeds are like this. The wrinkle shaped may be like this. Next, come to the seed color. Here again two traits. Seeds are in two colors. The one is yellow, another one is green. So here itself I tell you, so which trait is dominant, which trait is a recessive one. Here the round seed, I mean round shape is dominant. The wrinkle shape is recessive. Then yellow is the dominant, yellow color is the dominant, so green color is the recessive. So you have to remember this. So these are the two characters selected by the Mender. So with these two pair of opposing traits or contrasting traits. Next, so I tell you theoretically, so how the dihybrid cross was made. So after that we go for understanding of this cross with the help of the Punnett square. The first step, selection of parents, that is parent generation. Here two plants are selected. So one plant is round shaped seeds with yellow color. Then another plant is wrinkle shaped seeds with green color. So we can label first plant as the female plant, the second plant as a male plant for our understanding. First, it is necessary to write gametes. Gametes produced by these two parents. This parent produces gametes with RY. So here itself I wrote the genotype of this one. Capital R, capital R, capital Y, capital Y. So this is a small r wrinkled, small y represents the green color. Based on this genotype we can easily write the gametes. This plant produces gametes with small r, small y antennas. Now do the cross hybridization. Next step is hybridization, that is cross pollination is done here. This type of pollination, cross pollination. So due to the cross pollination, so we get first generation, that is F1 generation or first filial generation. So whatever may be the term, so you can use here. First filial generation. All the plants produced in the F1 generation so becomes heterozygous round yellow seeds produced plants. Means so capital R, small r, capital Y, small y. So this is the genotype of offsprings produced in the F1 generation. So we can tell this plant as heterozygous round seeds, yellow color seeds produced plant. So how we wrote this one? So here I do small rough work here. So capital R, capital Y, small r, small y, right? This capital R allele combined with this one. So capital R, small r. So this one combined with this one, capital Y, small y, capital Y, small y. Next step, selfing of the F1 generation plants. Now I take these two plants here, capital R, small r, capital Y, small y, into the same plant with same genotype, capital R, small r, y, capital Y, small y. Next, so write the gametes here. Then then it will be easy for you to find out the phenotype and genotype in the F2 generation. Gametes. So each plant produces 4 types of gametes. So one is capital R, capital Y. The another one is capital R, small y, then small r, capital Y. Another gametes containing small r, small y. Right? So this plant also produces the same gametes. Capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. See here, I will show you the rough work of this one. Capital R, capital Y, small y into capital R, capital Y, small y. Right. Capital R, capital Y, gametes. So here the any allele can combine and join with any other allele. Right? This is the segregation, law of segregation. So we saw the same thing. So occurs here. Then capital R, this allele also may fuse with this one. Then gametes become capital R, small y containing gamete. Then smaller, this smaller may combine with capital Y. Then gamete become smaller capital Y. So this smaller also have chance to fuse with this one. So smaller, small y. So these are the gametes. So match with these gametes. So this is the easy process. So to write the gametes. So 100% with 100% accuracy without confusion. Right. So now do the selfing. Right. Right, then F2 generation we get second filial generation. So we get. So theoretically I write there are totally 4 types of combinations. So we got in the F2 generation. The first one is round shaped yellow color seeds producing plants. Secondly, round shaped green colored seeds producing plants. Thirdly, wrinkled shaped yellow color seeds producing plants. Then finally, wrinkled shaped green color seeds producing plants. So these are the 4 types of plants or 4 types of combinations we Mendel got in the F2 generation. Here we need to make a keen observation, that is the round yellow, the first type, then last one wrinkled green, they are the parental types, right? This one and this one are the parental types. But so middle two, this round green then wrinkled yellow are the new combinations. They are not parental type, that is new combinations. So from this observation, so Mendel gave the conclusion. So this is the theoretical explanation of monohybrid cross. So let us let us see the receiving of the 4 varieties of combinations in the F2 generation diagrammatically, that is with the help of the Punnett square. So now I erase this one. So I draw the Punnett square this side. So it is very simple. So let me erase a little of this portion. So draw the Punnett square. So totally, so we should draw 16 squares. 4 into 4, 2, 3, then 4. Again 1, 2, 3, then 4. So it is very simple to write the gametes. On one side, so I wrote gametes of this plant, that is female plant, vertically. We always write the gametes of female plant, so vertically. So write these gametes, capital R, capital Y, capital R, small y, then small r, capital Y, then small r, small y. So these are the gametes of a female pair. Next, so on other side, horizontally, so we write the gametes of male, same gametes, capital R, capital Y, capital R, small y, small r, capital Y, then small r, small y. So these are the probabilities. So any gamete can so combine with any other gametes to give the either parental generation or new combinations. So let us write. So fuse this gamete with this one. The genotype of plant would be capital R, capital Y, capital Y, capital Y. Next, do the same with this gamete. The capital R dominant, then capital Y, small y. The dominant alphabet can always write first, then recessive next. Then do the same crossing with this gamete also. The capital R, small r, the capital Y, capital Y. Then so again with the same this gamete. The capital R, small r, capital Y, small y. Then so do the same. So follow the same process for these crossings. This gamete with this one. Then so fill this gamete with this one, and this one. So do the same for all these. Then capital R, capital Y, small y. Then capital R, small y. Then capital R, small r, capital Y, small y. Then capital R, small y. Then come to this gamete. So capital R, small r, capital Y. Then capital R, small r, capital Y, small y. Then small r, capital Y. Then small r, capital Y, small y. Then capital R, capital Y, small y. Then capital R, small y. Then small r, capital Y, small y. Then finally small r, small y, small y. So in this way we have to write the genotype. First write the phenotypes, right? First write the phenotype of these plants. So RR, YY, round yellow seeds producing plant. Here round again yellow seeds producing plant. Then dominant is R, then round. So here Y is dominant, then again round yellow plant. So here this is also round because R is the capital dominant. So Y is the dominant, again yellow, round yellow plant. Then come to this one. So capital R, round, then capital Y is there, then yellow. Yellow is the dominant color, round shape is the dominant shape. Then here also round, but both are recessive, small y. So this recessive represents the green color, round green color. Here round, so capital Y is there, so yellow. So here capital R, round, small y are there, then again green. So here round, capital Y, so yellow. So here capital R round, then capital Y yellow. So here small r recessive, so wrinkled, right? Wrinkled, so color is white, dominant, so yellow. So here small r again responsible for wrinkled shape, so capital Y responsible for yellow color, wrinkled yellow plant. And here capital R round, then capital Y, so capital Y is a yellow plant. Here R round, small y represents recessive, both are recessive, green. Then smaller wrinkled, capital Y yellow. Then smaller small y, both the pairs are in recessive condition, the plants become wrinkled shaped green color seeds producing plant. Now so everything is clear about phenotype of plants. So let us count the number of these four combinations. So I erase this portion. Right. The first combination, round yellow. Second combination round green. The third combination wrinkled yellow. The fourth combination wrinkled green. How many round yellow plants are produced here? Round yellow 1, round yellow 2, round yellow 3, round yellow 4, round yellow 5, then round yellow 6, then round yellow 7, then round yellow 8, then round yellow 9. So totally how many round shaped yellow color seeds produced plants are produced? Totally 9. Then come to round green plants. Round green, here is 1, then round green, here is 2, then round green, here is 1, 3. So totally 3. Next come to wrinkled yellow plants. Wrinkled yellow, here is 1, here wrinkled yellow second, then here wrinkled yellow 3. And again 3 plants were produced. The finally wrinkled green, only one remains. So only one remains. Here this 9:3:3:1 becomes the phenotype ratio of dihybrid cross. 9:3:1, right? Next, what is the genotype ratio of this dihybrid cross? Genotype ratio of this so this one. So these are the genotypes. Based on the genotypes, we have to write the numbers. So capital R, capital Y. So out of 16, totally 16 are there, right? Totally 16 plants are there. Out of 16, only one is there. Then capital R, capital Y, small y. So how many are there? Here is 1. So here is 1. So then 2. Then capital R, small r, capital Y. Here is 1. then here is 1 2, right, only 2, then R Y Y, capital R small r, capital Y small y, 1 2 3 4, totally 4. Next capital R, capital Y small y, so 1, only 1. Next capital R, capital R, capital Y, capital Y, but where it is, capital R, small y, small y, 1 capital R, small y, where it is, 2. Next this one, capital R, capital Y, small y, here is 1, here is 1 2, so it was finished already. So finally, so we got 1 in this ratio, right? The genotype ratio of dihybrid cross is 1:2:2:4:1:2:1:2:1. So this is the genotype ratio of dihybrid cross. Next, the conclusion of dihybrid cross. So generally here one product rule can be applied, right? Product rule can be applied for getting the phenotype ratio. What is product rule? By multiplying monohybrid cross phenotype ratios, so we can get the phenotype ratio of dihybrid cross. That is called product rule. For example, 3:1 is the phenotype ratio in the monohybrid cross. Then multiply with the same. So 3:3 9, 3:1 3, 1:3 3, then 1:1 1. So 9:3:3:1 is the phenotype ratio of the dihybrid cross. Here the 3:1, so is the proportion of type of plants we get in the dihybrid cross. So already I mentioned the 4 types. One is round yellow color plants, then round green color plants, then wrinkled yellow. The fourth one is wrinkled green. So these plants got in the ratio of 3:1, 3:1, 3:1. So here we know the parent type. The two new combinations are, one is round yellow. This is the parental type. The second one is round green. It is a new combination. The third one is wrinkled yellow. This is also new generation. The last one is wrinkled green, pure recessive. It is the parental one. So based on this observation, so what Mendel concluded is any pair of contrasting traits do not interfere with the other pair of contrasting traits. So any pair can combine with any other pair. That is called a independent assortment. That is called a independent assortment. Independent assortment. So it is about the complete picture of dihybrid cross did by the mentor. So thank you for watching.